Hey students, today we are going to start with the book VCE Biology, and we are going to read chapter number 5, photosynthesis, and we'll be doing 5A in this audio, the process of C3 photosynthesis. Let's get started. 5A, the process of C3 photosynthesis. Overview of C3 photosynthesis. Overview. Photosynthesis is the biological process where photoautotrophs capture light energy from the sun and convert it into chemical energy. Theory details. Overview of photosynthesis. Plants are photoautotrophs, meaning that they do not consume the food they need to survive as we humans and other animals do. Instead, they create their own energy via photosynthesis. Photosynthesis is the process in which light energy is harnessed to produce glucose, the energy source of plants. Algae and photosynthetic cyanobacteria are also photoautotrophs capable of undertaking photosynthesis. In a sense, photosynthesis uses two inputs, carbon dioxide and water, to produce the outputs glucose, oxygen, and water. Figure 1. For this process to occur, sunlight is also required to energize the reaction. Glucose is the primary product of photosynthesis. It is either used immediately as a source of energy for cellular respiration, stored as starch, or used to form more complex molecules such as cellulose. Photosynthesis is not as simple as figure 1 makes it look. Instead, it is a complex series of biochemical reactions and their regulatory enzymes that can be broken into two stages, which we will explore later in this lesson. Photoautotroph, an organism capable of undertaking photosynthesis. Photosynthesis is the process of capturing light energy to power the production of glucose and oxygen from carbon dioxide and water. Plant structures involved in photosynthesis. Leaves are the main site of photosynthesis in plants and typically have a large surface area to maximize the amount of light hitting the surface. Leaves contain many different cell types that perform varying functions, from structural support to channeling water and nutrients. The main cells in leaves that photosynthesize are called mesophyll cells. Inside mesophyll cells are large populations of chloroplasts, the organelle that is the site of both stages of photosynthesis. To zoom in ever further, within chloroplasts is the photosynthetic pigment known as chlorophyll, which is directly responsible for initiating photosynthesis by capturing and being energized by light energy. Tiny pores on the surface of leaves, known as stomata, open to allow carbon dioxide in the atmosphere to diffuse into the leaf. Stomata can also close to prevent water loss from the leaf in dry conditions. Water is absorbed by the root hair cells of plants from the soil and transported through the xylem to photosynthesizing cells. Mesophyll cell, a plant cell type found in leaves that contain large amounts of chloroplasts. Chloroplast, a membrane-bound organelle only found in plant and photoautotroph cells, that is the site of photosynthesis. Chlorophyll, a chemical found in the thylakoid of chloroplast. It is responsible for absorbing light energy in photosynthesis. Stoma, plural stomata, a small pore on the leaf's surface that opens and closes to regulate gas exchange. Xylem, vascular tissue in plants responsible for transporting water and minerals from the roots to the leaves. The light-dependent stage. Overview. In the first stage of photosynthesis, the plants are dependent on light to split water into hydrogen and oxygen. This light-dependent stage occurs on the thylakoid membranes of chloroplasts. Theory details. As the name suggests, the light-dependent stage of photosynthesis only occurs when light is present. The light-dependent reactions occur on the chlorophyll-filled thylakoid membranes, which make up the grana inside a chloroplast. Figure 4. The reactions in the pathway are catalyzed by various enzymes, and the purpose of this first stage is to generate the high-energy coenzymes NADPH and ATP to power the second stage of photosynthesis. The inputs of the light-dependent stage are 12 water molecules, 12 NADP positive, 18 ATP plus inorganic phosphate. The outputs of the light-dependent stage are 6 oxygen molecules, 12 NADPH, and 18 ATP. Light-dependent stage. The first stage of photosynthesis where light energy splits water molecules into oxygen and hydrogen inside the thylakoid membranes, also known as the light-dependent reactions. Thylakoid, a flattened sac-like structure housed inside the chloroplast. Each thylakoid is made up of a chlorophyll-containing membrane enclosing a lumen. Thylakoids are the location of the light-dependent stage of photosynthesis. Grana, plural grana, a stack of thylakoids. NADPH, a coenzyme that is a proton and electron carrier in photosynthesis. ATP, adenosine triphosphate, a high-energy molecule that, when broken down, provides energy for cellular processes. The steps in the light-dependent stage are: number 1. Inside the thylakoid, light energy excites electrons in chlorophyll. The excited electrons move along proteins in the thylakoid membrane. As they move, the energy in the electrons powers the pumping of hydrogen ion into the thylakoid lumen. Water donates electrons to chlorophyll to replace the electrons that leave, which causes water to split into oxygen and 2 hydrogen ions. This process is known as photolysis. Photolysis, the process in which molecules are broken down by the action of light. Number 2. The oxygen is released from the chloroplast. It will either diffuse out of stomata or into the environment, or be used as an input for aerobic cellular respiration. You will learn more about this in Chapter 6. Number 3. The hydrogen ions from water molecules are used to generate the high-energy coenzyme NADPH. From the reaction, NADP positive plus H positive gives you NADPH. The movement of hydrogen ions down its concentration gradient, maintained by energy from excited electrons in step 1, generates the high-energy coenzyme ATP. From the reaction, ADP plus inorganic phosphorus gives you ATP. Figure 7. Number 4. ATP and NADPH coenzymes then move on to the light-independent stage. Ultimately, these steps demonstrate how the thylakoid turns 12 water, 12 NADP positive, and 18 ATP plus inorganic phosphorus into 6 oxygen, 12 NADP, and 18 ATP. In summary, during the light-independent stage of photosynthesis, sunlight excites an electron within chlorophyll. Water absorbed by a plant's root hairs is split into oxygen and hydrogen ion as it donates 1 electron to the chlorophyll. The excited electron and hydrogen ion from water lead to the production of the coenzymes NADPH and ATP. The oxygen is released out of the chloroplast and the coenzymes are ready for the second stage of photosynthesis. Enzymes and coenzymes in photosynthesis. Enzymes catalyze most of the reactions in photosynthesis. For example, in the light-dependent stage, the enzyme ATP synthetase catalyzes the reaction ADP plus inorganic phosphorus to form ATP using energy from the flow of hydrogen ions down its concentration gradient. Having enzymes regulate each step in photosynthesis ensures reactions are sped up and controlled, so plants can metabolize efficiently. We will focus on the function of another key enzyme in photosynthesis called rubisco in the next lesson. The coenzymes NADPH, and ATP cycle through both stages of photosynthesis. In the light-dependent stage, the NADP positive and ADP plus inorganic phosphorus inputs are turned into high-energy NADPH and ATP outputs. These two high-energy molecules then go on to be inputs for the light-independent stage. As you will soon see, the coenzymes donate their energy in the light-independent reactions, forming the unloaded NADP positive and ATP plus inorganic phosphorus. The unloaded coenzymes then return back to the light-dependent reactions and the cycling continues. Figure 8. The light-independent stage. Overview. During the second stage of photosynthesis, glucose is produced from carbon dioxide, NADPH, and ATP through a cycle of reactions occurring in the stroma of chloroplasts. Theory details. Unlike the light-dependent stage, the light-independent stage of photosynthesis does not require light to occur. Instead, the reactions are energized by the ATP and NADPH coenzymes produced in the light-dependent reactions. The light-independent stage occurs in the stroma, is facilitated by enzymes, and cycles through multiple reactions, which is why the stage is also referred to as the Calvin cycle. The inputs of the light-independent stage are 6 carbon dioxide molecules, 12 NADPH, and 18 ATP. The outputs of the light-independent stage are glucose, C6H12O6, 6 water molecules, 12 NADP positive, and 18 ADP plus inorganic phosphorus. Light-independent stage, the second stage of photosynthesis where carbon dioxide is used to form glucose in the stroma of a chloroplast. Also known as the Calvin cycle, the dark stage, or the light-independent reactions. Stroma, the fluid substance that makes up the interior of chloroplasts. It is the site of the light-independent stage of photosynthesis. The steps in the light-independent stage are: number 1. Carbon dioxide molecules enter the Calvin cycle and undergo initial reactions. During these changes, the carbon from carbon dioxide combines with a 5-carbon molecule, then splits into 2 into 3 carbon molecules, which continue along the cycle. Number 2. NADPH molecules formed in the light-dependent reactions donate their hydrogen ions and electrons, and ATP molecules break into ADP and inorganic phosphor to release energy to facilitate further changes to the carbon molecules. Number 3. Carbon molecules continue to change and rearrange as they move around the cycle. Eventually, one specific 3-carbon molecule is created and leaves the cycle, going on to contribute to the formation of glucose. Overall, 6 carbon dioxide molecules must enter the cycle to produce glucose. Carbon dioxide is equal to 1 carbon and glucose is equal to 6 carbons. Number 4. Some of the oxygen molecules left over from the breaking of carbon dioxide at the beginning of the cycle combine with hydrogen ions from NADPH to create the output water. Overall, within the stroma, the reactions turn 6 carbon dioxide, 12 NADPH, and 18 ATP into C6H12O6, 12 NADP positive, 18 ATP plus inorganic phosphorus, and 6 H2O. To summarize, the light-independent stage of photosynthesis, carbon dioxide collected from the stomata in leaves enters a cyclic reaction. The carbon from carbon dioxide undergoes reactions powered by ATP and NADPH to produce a series of carbon-based molecules. Eventually, a specific carbon molecule is reached that goes on to contribute to the formation of glucose, with water also being produced in this stage. The production of glucose is the main outcome of photosynthesis. The plant, through a series of reactions over the 2 stages, has now converted sunlight energy into chemical energy stored within the bonds of a glucose molecule. The glucose is transported out of the chloroplast for cellular respiration or conversion into complex carbohydrates. Theory summary. Photosynthesis is the process of turning light energy into chemical energy in the form of glucose. Photosynthesis occurs in two distinct stages, summarized in Figure 12 and Table 1. There are a number of factors that would make growing plants on Mars extremely difficult. Mars is about 80 million kilometers further away from the Sun than Earth, so there is far less light energy reaching the surface of Mars. Additionally, Mars experiences frequent dust storms and strong winds and thus further obscure the Sun. Plants that we try to grow on Mars surface would not receive enough sunlight to undertake the light-dependent stage of photosynthesis and as a result would die. For more of these reasons and more, scientists have been focused on researching artificial greenhouses that could be implemented on Mars where the light and climate of a plant's environment can be controlled. So this was 5A. Thank you.